This is our research-grade upright fluorescence microscope. Let me introduce its components from top to bottom. On the top it is a microscope camera. Connects to the computer through a USB cable, and it contains microscope imaging analysis software. Allows preview, take photo, record video and others. Here we can see its performance image and software on the screen. Under the camera, there is a C-mount adapter, used to connect the microscope with camera. It has different models in different zoom times. 4 different sizes of camera sensor to show field of view as wide as possible. Then let us see the trinocular observation head. There are 2 eyepieces on it. The most biggest field of view we can do at 25 micrometers. More area of sample can be seen. It improves lab working efficiency. Now let us move to the fluorescence attachment. In front end of it is a wheel with filter cubes and filters inside, which used for fluorescence. At the end of it is a multi-channels LED light source. It covers most lighting wavelengths of common used fluorescence. The LED light source can be instant on and off. That is more convenience than mercury light source. What is more, it is long working life, saves energy and safety to use, and save cost for long time using. The most important, LED is cold light source. Less heat comes out. That is friendly to the sample we study. In the middle we can see the frame of the microscope. The bright field light source is built in the frame, and there is a power button. On top of the frame is a nosepiece. It used to hold objectives, and we have built-in code to that nosepiece. It can be integrated with the software to control and recognize the nosepiece. It allows to call the matched calibration automatically. It reduces your working progress and save time. Under the objectives, it is a mechanical stage. By international standard, we sure its smooth working and stability. Keep moving down, we can see the condenser. On the bottom, it is the transmitted bright field illuminating system. Then we can see 2 hand wheel on both sides of the microscope. They are used for focusing. By control the lifting of the stage, you can lock the focusing knob when it arrives the position you want, and adjust its tension as your using habit. Now I take a slide to show how is the bright field and fluorescence. The slide I am putting on is a pathology slide. It is tissue of heart which is commonly used in pathology, and a slide of stomach wall. Their staining usually are rich. Now we can see they are stained of HE. Live view is very smooth when moving the slide. Let us move the objective to 40 times. After changing objective, you need to do fine focusing. Adjust the bright field light intensity to the suitable brightness. Compare its photo performance with eyepiece. Now we can see how its performance. Also, we can zoom out the photo by software to check details. You can draw the photo to see any area you want to. It is still very clear even zoom out. Next, we can check its fluorescence performance. In my hand, it is a digital control box of fluorescence light source. Press the power button behind it to light on. The RGB UV red, 4 channels fluorescence light sources. The light intensity of each channel are different, and each channel can be controlled separately to open and close and adjust brightness. That is an advantage for quantify, which mercury cannot do. What we see now is Doppler excited by UV light source. The signal of Doppler is usually very strong. We can see the light intensity of UV light is only 18%. We also can get down its brightness by setting exposure time. Now we have got a good fluorescence. Move to the green filter to see red color fluorescence. Because every channel is suitable, light intensity actually is different. Such as red fluorescence signal intensity is kind of weaker. It can be adjusted by software to get a more pure photo, commonly by setting darker background. Tiny fine focusing to make it sharper. Let us take a photo. We can see each channel fluorescence are very vivid.